Welcome to the Super Simple Science Show. Today we'll continue our Intro to Chemistry series with an episode on aerobic myoglobin biocatalysis and dipeptidic solvents. To start us off, I'll send you my co-host, Kaylee.
Biocatalysis uses biological enzymes or whole cells to catalyze chemical reactions. A common biocatalyst you might be familiar with is baker's yeast. This specialized fungus converts glucose into ethanol and carbon dioxide and is a vital component in bread and beer production. Biocatalysts are a handy tool for chemists. They are highly selective to only a few substrates, offer fast and efficient yields in complex reactions, require mild conditions, and are renewable and environmentally conscious. Our research focuses on one specific biocatalyst called myoglobin.
Myoglobin is a heme-containing protein that stores oxygen in muscle tissue. For our experiments, we used mutated sperm whale myoglobin, synthesized in specialized E. coli cells. Over several days, we cultured, induced, and lysed cells before using various purification techniques to collect a pure myoglobin solution. Myoglobin has a high affinity for atmospheric oxygen, reducing its viability as a biocatalyst. It will readily perform reactions in an anaerobic environment, but the additional time and effort required makes it less than ideal for industrial applications. To circumvent this problem, we explored a designer solvent class called dipeptidic solvents, or DES. Our resident DES expert, David, is here to tell you all about it.
Dipeptidic solvents are tunable solvents formed from mixing hydrogen bond acceptors and donors in a certain molar ratio. Many hydrogen bond acceptors and donors, such as choline chloride and urea, start as solids, but upon mixing, the change in intermolecular forces lowers the melting point below room temperature and results in a solvent with many favorable properties. The polarity of these solvents are compatible with various enzyme and substrate classes, and we could change hydrogen bond donors and acceptors to create a solvent specific to an enzyme's needs. These solvents are safe for the environment, easy to prepare, and cost-effective. Past research has focused on choline chloride as the hydrogen bond acceptor in the DES, and these solvents have proven to be a stable environment for our enzyme. This semester, I've been working to determine enzyme stability in betaine-based DES, while Kaylee and Mason have continued research on enzyme function.
My research initially focused on a cyclopropionation reaction with styrene and ethyl diazoacetate. However, after countless hours in the lab and a series of failed reactions, I began to study two reactions with thiophenol and allyphenol sulfide. At the time, we could not determine the extent of the reaction from gas chromatography, but the initial results were promising. Thankfully, Kaylee's initial substrate, aniline, was highly successful.
So far, the most successful choline chloride-based DESs were glycerol and xylitol, which gave percent yields from 50 to 70%. For betaine and water-based DES, xylitol and sorbitol were clear winners with percent yields from 65 to 80%.
Thanks for watching the Super Simple Science Show. We hope you learned something. Take care, and we'll see you on the next one.